Hello friends, welcome to the second lecture on mechanism of action of antibiotics. In the first lecture, we learn about history, discovery, classification and the general overview of bacterial infection. In the classification, we learn that antibiotics can be classified into 5 subgroups based on the mechanism of action. Today, in today's lecture, we will discuss one by one mechanism of action in details along with the selective toxicity and other effects of the antibiotics. So the 5 classes were cell wall synthesis inhibitors, protein synthesis inhibitors, nucleic acid synthesis inhibitors, metabolic pathway inhibitors, and lastly the cell membrane disruptors. Starting from the cell wall synthesis inhibitors, which include penicillins, cephalosporins, and carbapenems. So mechanism of action is basically by the 6 methods. First one is by targeting peptidoglycan synthesis. The bacterial cell wall is made up of peptidoglycan, a polymer composed of sugar and amino acid chains that are cross-linked to provide strength and rigidity. Cell wall synthesis inhibitor act specifically during the synthesis and cross-linking of these peptidoglycan strands. The second way of action is interference with transpeptidase enzyme. Penicillin, cephalosporins, and carbapenems that are beta-lactam antibiotics share a beta-lactam ring structure. This structure enables them to bind to the penicillin binding protein PBP, which include enzyme like transpeptidase. Transpeptidase is responsible for cross-linking of the peptidoglycan chains by forming peptide bond. By inhibiting this enzyme, these antibiotics prevent the cross-linking and that result in a weak and unstable cell wall. Third is the bactericidal effect. Without cross-linking, the peptidoglycan layer becomes fragile and unable to withstand the osmotic pressure inside the bacterial cell. As a result, the bacterial cell undergo lysis, that is, the cell ruptures and dies, making these drugs bactericidals. Then specific drugs in each class are penicillins, binds to protein binding peptidoglycans, disrupting the cell wall synthesis and causing bacterial lysis. Variants of penicillins like amoxicillin and methicillin have different spectrum of activity due to their chemical modifications. Whether the cephalosporins work similarly to penicillin but are more resistant to beta-lactamase enzymes, which can degrade penicillins. Cephalosporins are classified into generations based on their spectrum of activity, like first generation, second generation, third generation, fourth generation, and novel cephalosporins, that is fifth generation cephalosporins. Carbapenems are the broad spectrum beta-lactam that inhibit PBPs and are highly resistant to beta-lactamases, making them effective against the multi-drug resistant bacteria. Then the next step is autolysin activation. These inhibitors can also trigger the activation of bacterial autolysins. Autolysins, the enzymes that degrade the cell wall. Normally, autolysins help remodel the cell wall during growth, but their uncontrolled activity in the presence of beta-lactam antibiotics accelerate the cell death. Then selective toxicity, like why these are not acting on human cells? Because the cell wall synthesis inhibitor are selective for bacteria, because human cell lack a cell wall and associated enzymes. This makes them safe for human use. Next class is protein synthesis inhibitors. Protein synthesis inhibitors are the antibiotics that target bacterial ribosome, disrupting the process of translation and thereby preventing the production of essential proteins required for bacterial survival and growth. Here we will discuss the detail of their mechanism. So mechanism of action starts with the binding to ribosomal subunit. Protein synthesis in bacteria involve two ribosomal subunits. As you know, 30S and another one is 50S. These subunits work together to translate messenger RNA, that is mRNA, into the polypeptide chains. Protein synthesis inhibitor act by binding to one of these subunits and interfere with the different steps of translation. Like tetracyclines bind reversibly to the 30S ribosomal subunit. Specifically, they prevent the attachment of amino acyl tRNA to the A site of the ribosome. halt the addition of new amino acid to the growing polypeptide chain, which lead to a bacteriostatic effect, meaning the drugs inhibit bacterial growth without directly killing the cells. Another class in the protein synthesis inhibitors is macrolides. Macrolides bind to the 50S ribosomal subunit, specifically targeting the region that facilitate peptide chain elongation. They block the translocation step during translation, where the ribosomes shift along the mRNA to add new amino acid. This telling prevents the formation of complete polypeptide and exert a bacteriostatic effect. The third class is aminoglycoside. Aminoglycoside binds irreversibly to the 30S ribosomal subunit, which lead to two critical disruptions. First disruption is misreading of mRNA, which result in the production of faulty or non-functional protein. Then second disruption is premature termination of translation, which lead to incomplete polypeptides. Unlike tetracycline and macrolides, aminoglycosides are bactericidal because of the misfolded protein disrupt bacterial membrane integrity and essential functions. So, overview of these three classes, tetracycline, macrolides, and aminoglycoside. The tetracycline reversibly bind to the 30S ribosomal subunit. Macrolide bind to the 50S ribosomal subunit. And aminoglycoside irreversibly binds to the 30S ribosomal subunit. So the specific action on translation steps are: each class of protein synthesis inhibitor act on different phases of translation. So initiation blockers. Some drugs may prevent the initiation of protein synthesis by interfering with the assembly of ribosomal subunit, mRNA, and the initial tRNA. Then the elongation inhibitors. Most drugs, likely tetracycline, macrolides, these disrupt the elongation phase by targeting the attachment of amino acid or the movement of the ribosome. Third is termination impairment. Few drugs interfere with the proper termination, although aminoglycosides indirectly affect this step, though misreading of mRNA. Then why these protein synthesis inhibitors does not affect the human ribosome? The answer to this question is there is a difference between bacterial and human ribosomes. So bacterial ribosome is 70S, consists of 30S and 50S subunit, whereas human ribosome 80S, it consists of 40S and 60S subunit. These structural difference ensure that these antibiotics selectively target bacterial translation without significantly affecting the human protein synthesis. The third group is nucleic acid synthesis inhibitors. These are a class of antibiotics that disrupt the production of DNA and RNA, essential molecules for bacterial replication and transcription. These drugs specifically target bacterial enzymes involved in nucleic acid synthesis, leading to the inhibition of growth or bacterial cell death. So we will explain this by subclasses of under this class, that is nucleic acid synthesis inhibitors are: first one is quinolones. Quinolones, including fluoroquinolones, are potent inhibitor of bacterial DNA replication. They achieve this targeting key enzymes involved in the supercoiling and uncoiling of DNA, which are the critical needs for the replication of DNA. So DNA gyrase, that is found in gram-negative bacteria, and topoisomerase 4, that is found in gram-positive bacteria. These are two target sites of these quinolones. Quinolones stabilize the enzyme DNA cleavage complex, prevent the enzyme from resealing breaks in the DNA strand after unwinding. This results in double-strand DNA breaks, disrupt replication, and leading to the bacterial cell death. These drugs are bactericidal, kill the bacteria irreversibly, damage their DNA. Second class in the nucleic acid synthesis inhibitor is rifamycins. Rifamycins such as rifampin inhibit bacterial RNA synthesis, blocking transcription. The target for rifamycins are RNA polymerase, specifically the bacterial beta subunit of the enzyme. Rifamycin bind to the RNA polymerase and prevent the initiation of mRNA synthesis. This halt the production of protein needed for bacterial survival and replication. As outcome, rifamycins are bactericidal against many bacteria, including Mycobacterium tuberculosis, making them critical in the treatment of tuberculosis. So the selective toxicity of quinolones and rifamycins are: because both exploit structural differences between bacterial and eukaryotic enzymes. The quinolones, DNA gyrase, and topoisomerase 4 are unique to bacteria and absent in human, ensure the selective action of these quinolones. Then rifamycins, the bacterial RNA polymerase differ from human counterpart, enable selective inhibition. The clinical significance of quinolones, these are used for treating a wide range of infection, including urinary tract infection, UTIs, respiratory infection, and gastrointestinal infections. Rifamycins, whereas primarily used against the Mycobacterium species in the disease like tuberculosis and leprosy. The fourth group is metabolic pathway inhibitors. Metabolic pathway inhibitors target specific biochemical pathway essential for the bacterial growth and survival, particularly those involved in the synthesis of nucleotides and folic acid. These groups exploit the fact that bacteria synthesize folic acid de novo, that is from existing, whereas humans folic acid de novo means new, whereas the human obtained it from the dietary sources. Here, so we will discuss the subclasses. Sulfonamides. Sulfonamides are the structural analogues of para-aminobenzoic acid, PABA, PABA, that is a precursor of synthesis of folic acid. Sulfonamide competitively inhibit the enzyme dihydroteroate synthase, which catalyze the conversion of PABA into dihydroteric acid and intermediate in folic acid synthesis. By blocking this step, sulfonamide prevent the formation of folic acid, which is required for the synthesis of purines and pyrimidines, that are the basically building blocks of the DNA and RNA. As outcome, the sulfonamides are bacteriostatic, meaning that they halt bacterial growth by starving the bacteria of nucleotides needed for replication and the repair. So second subclass is trimethoprim. The trimethoprim specifically inhibit dihydrofolate reductase, the another key enzyme which is responsible for the folic acid synthesis. Dihydrofolate reductase convert dihydrofolate into tetrahydrofolate, the active form of folic acid which is required for nucleotide synthesis. By inhibiting this enzyme, trimethoprim block the final step in the folic acid production, synergistically enhancing the effect sulfonamides when used together. As an outcome, trimethoprim is also bacteriostatic, and when combined with sulfonamide, as in combination drug like cotrimoxazole, these two drugs exhibit synergistic effect, leading to increase in antibacterial efficacy. Now, coming to the selective toxicity of this class, that is metabolic inhibitors. So why this does not affect the human? Let's understand the difference between bacteria and humans. So bacteria require folic acid synthesis for survival and replication, whereas humans do not synthesize the folic acid. Instead of that, they obtain it from the dietary source. As a result, these drugs selectively target bacterial pathways without harming human cells. It means when suppose any bacteria is infecting a human, then human has stored folic acid, but bacteria cannot utilize directly the folic acid. They have to synthesize it. Okay, so these two drugs, sulfonamide and trimethoprim, block the folic acid synthesis in the bacteria. Clinical significance of sulfonamides and trimethoprim are urinary tract infection, often treated with cotrimoxazole. Then respiratory tract infections, cotrimoxazole is also effective against the type of pneumonia such as pneumocystis jirovecii pneumonia. Other bacterial infection including caused by the E. coli, Klebsiella and Enterobacter species. And the last class, that is fifth class, is cell membrane disruptors. Cell membrane disruptors are the antibiotics that directly damage the bacterial cell membrane, compromising its integrity and leading to cell death. These drugs are particularly effective against gram-negative bacteria, which have an outer membrane in addition to their inner cell membrane. So under this group, the polymyxins and daptomycins are two classes. So polymyxins such as polymyxin B and polymyxin E, that is colistin. These target the outer membrane of gram-negative bacteria. They specifically interact with lipopolysaccharide, that is LPS, which is a major component of outer membrane of gram-negative bacteria. So the polymyxins are cationic peptides, that is are positively charged, that bind to the negatively charged phosphate group in the lipopolysaccharide layer. This binding displaces divalent cations, that is magnesium, calcium, etc., that normally stabilize the outer membrane. So the disruption of lipopolysaccharide causes disorganization and increase the permeability of outer membrane. Polymyxin also insert themselves into the inner cytoplasmic membrane, forming pores and cause the structural damage to the membrane. As an outcome, the bacterial cell loses essential ions and molecules, leading to the leakage of cellular contents and ultimately cause the osmotic imbalance, membrane lysis, and bactericidal effect. The another class in this is daptomycin. So the daptomycin target the cytoplasmic membrane of gram-positive bacteria, make it unique among the cell membrane disruptors. So daptomycin bind to the bacterial membrane in a calcium-dependent manner. So it oligomerize that is oligomerize means forming the complex on the membrane surface and insert into the lipid bilayer. This insertion disrupt the membrane potential by forming ion conducting channels. Then these channels lead to the leakage of ions like potassium, which result in membrane depolarization, and as a result, integrity of the membrane is lost. So the disruption of membrane potential halt the essential cellular process like ATP synthesis, which result in the bacterial death, making daptomycin bactericidal. So polymyxin are gram-negative specific and daptomycin is gram-positive specific. So the same question arises here: why these does not affect the human? So the polymyxin exploit the unique composition of the bacterial outer membrane such as lipopolysaccharide, which is absent in human cells. So that thing is not present in human, so it can't affect the humans. So another class, the daptomycin, which target the bacterial membranes with specific lipid composition that is also not found in the human cell membrane. However, the polymyxin can have nephrotoxic and neurotoxic effect in human because they may interact with the lipid component of the mammalian cell membrane at higher dose. So we discussed the mechanism of action of each classes, that is cell wall synthesis inhibitors, protein synthesis inhibitors, nucleic acid synthesis inhibitor, metabolic pathway inhibitor, and cell membrane disruptors. Also covered the subclasses under each category. So as summary, the mechanism of action for the categories of antibiotics, the cell wall synthesis inhibitors are include penicillin, cephalosporins, and carbapenemas, which disrupt the bacterial cell wall construction by inhibiting enzyme like transpeptidase, which prevent the cross-linking of peptidoglycan strand, weak the bacterial cell wall, and cause the cell lysis. Hence, these are bactericidal. Then protein synthesis inhibitors, these drug target the bacterial ribosome and block the translation process. Tetracycline reversibly bind to the 30S subunit of ribosome, macrolides to the 50S subunit, and aminoglycoside irreversibly bind to the 30S. In combination, these cause the where the tetracycline halt the elongation process, macrolides then inhibit the translocation, and aminoglycoside cause the mRNA misreading and generation or synthesis of faulty proteins. Then third class, nucleic acid synthesis inhibitors. These interfere with the DNA and RNA synthesis. The quinolones, which inhibit the DNA gyrase and topoisomerase 4, which cause the DNA strand to break and bacterial cell death. So rifamycin block the transcription by inhibiting the RNA polymerase. Fourth class was metabolic pathway inhibitors. In this, there were two subclasses, sulfonamide and trimethoprim. Sulfonamide compete with PABA and dihydropteroate synthase, hold the nucleotide formation. Whereas in the trimethoprim inhibit dihydrofolate reductase, which is the final enzyme required for the folic acid synthesis, and hence prevent the production of folic acid. Then last class was cell membrane disruptors. These antibiotics damage the cell membrane. In this, there were two subclasses, polymyxin and daptomycin. Polymyxin are gram-negative specific and daptomycin is gram-positive specific. Polymyxin bind to the lipopolysaccharide in the gram-negative, whereas the daptomycin insert into the membrane of gram-positive bacteria, forming the ion channels and disrupt membrane potential. So exhibit a bactericidal effect. So this was all about today's lecture. In the third week, we will discuss the spectrum of activity of antibiotics, that is narrow spectrum and broad spectrum antibiotics, which are the drugs under them, and how they act, that is a mechanism of action of different classes, what are the advantages, what are the uses of these narrow spectrum and broad spectrum, where to use, in which condition we use. So we will deal with all these details. Till then, if you like the video and the content, then share among your colleagues. Also comment your queries if you have, and also give suggestion if you want any changes in the way of presenting or way of video making. Then kindly comment in the comment box. It was all about today. So till then, take care. Thank you. Bye.